This video is part of the lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. The gene construct is a synthetic DNA sequence that will be inserted in the modified organism. It contains at least 4 elements. A promoter sequence, which indicates, initiates, and regulates the beginning of DNA transcription. One or several genes of interest coding for traits that the breeder wishes to improve. A reporter gene, which indicates whether the insertion of the gene construct was successful in the organism. A terminator sequence indicating where to stop the transcription of the construct. Here we will see in detail the roles of the reporter gene and how it is used to screen for successfully transformed plants. The reporter gene is coding for a gene product that is easy to detect either visually or through other screening methods. If a DNA construct is successfully inserted in the plant, the RNA polymerase will transcribe not only the RNA of the gene of interest, but also the RNA of the reporter gene. Both RNAs are then going to be translated into their corresponding products. While the trait of interest might not be directly detectable, either because it expresses only at a certain plant stage or because it requires special tools for its measurement, the reporter trait can be immediately seen or detected with a simple screening procedure. Imagine a gene of interest coding for a trait that is only expressed when the plant reaches the adult stage, such as sugar content of fruit. In a protoplast culture that has undergone a genetic transformation process, it is impossible to distinguish successfully transformed cells from those who weren't transformed at all. Without a reporter gene, the plants will need to first be grown until they produce fruit. Then the sugar content of their fruit needs to be measured in order to be able to identify the successfully transformed plants. All this requires a lot of time and resources to grow all these plants. Having reporter genes that are expressed early or immediately after a successful genetic transformation allows to select only the transformed cells for tissue culture, rather than investing in the growth of all cells without discriminating transformed from non-transformed cells. The reporter genes can be positive, coding for a trait that either enhances the plant performance or somehow distinguishes transformed from non-transformed plants. Such is the gas reporter system, which produces intense blue color if it is supplemented with a substrate, or fluorescent and bioluminescent genes. Reporter genes can be negative, such as herbicide or antibiotic resistance genes, in which case all but the transformed cells will be destroyed after applying a negative selection treatment. Finally, instead of using reporter genes, the transformed plants can be genotyped, using specific primers so that only the genes of interest are amplified. However, before genotyping transformed cells, they first need to be induced to multiply so that enough material is available for the genotyping assay, and also to make sure that the DNA which was not successfully inserted in the cell genome has been eliminated. One important issue, especially when using negative reporter genes, is the possibility of their transfer to other organisms via horizontal gene transfer. If you're eating a genetically modified plant that contains a reporter gene for antibiotic resistance, there is a very small but not impossible chance that the resistant gene is integrated in a bacterium from your intestinal flora, resulting in an antibiotic-resistant bacterium. To avoid this, a lot of effort is made for reporter genes to be removed after a plant has been successfully transformed. One way to remove a reporter gene is to use a more complex gene construct, in which in addition to the 4 basic elements, there are also restriction sites in between which are placed the reporter gene, a recombinase gene associated with an inducible promoter, which is activated after exposition to a chemical substance. After confirming the successful transformation of the plant via negative screening, the plant is exposed to a chemical activator that will initiate transcription of the recombinase. The enzymes produced by the gene will then recognize and bind to the restriction sites, effectively cutting out the reporter and recombinase genes from the genome. Then the cell's DNA repairing mechanism will take over to fix the broken DNA strand. In conclusion, reporter genes are used to indicate successful transformation of a genetically engineered organism. They are particularly useful if a quick screening of the transformation can be done immediately after it, without having to grow or multiply the transformed cells. Depending on the type of screening, reporter genes are either positive, such as GUS or fluorescent genes, or negative, such as antibiotic and herbicide resistance genes. There is a small but still present risk of horizontal transfer of the reporter genes from the genetically modified organism into other organisms, which can result in the emergence of antibiotic-resistant microorganisms or herbicide-resistant weeds. To avoid this, reporter genes are removed after they have accomplished their function in the genetic engineering process.